Good morning, everyone, and welcome to the third day of our online program to the introduction to microbiology and biotechnology, organized by the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. So, on the behalf of this department as well as from the university, I welcome you all to our third session. And today, again, we have a very exciting speakers and talks. Two speakers, eminent speakers, we do have with us today from our department. Professor Shomshubra Thakur Chaudhuri is going to deliver his lecture, and another speaker we have Dr. Mohit Mojumdar, and Professor Thakur Chaudhuri will be speaking on the central dogma of molecular biology, a basic insight. And Dr. Mohit Mojumdar will be speaking on the introduction to bioinformatics. So as I'm seeing that these two topics are very interesting and very much emerging areas of research in today's biotechnology and microbiological world. So I think you all will enjoy a lot for today's session and you will have a very good lesson, take home lesson from this session. So Professor Calcutawala, are you there?
I'm there, Rajat.
Yes. So, if everything is fine, if the live streaming, we have the YouTube live streaming also going on with us.
Yes,
So, if that is going on very nicely, so it is almost 9:57. 3 minutes we do have on our hands. So, in these 3 minutes, I'm just trying to, speaking about my partner. Um, for previous days I'm speaking about the same thing, but still, because maybe some participants are coming newly in this session, so for them, I'm just telling that our department is a very vibrant department and very, I mean, uh, active department in the beautiful campus of Sister Nivedita University, Kolkata. I, Dr. Rajat Pal, is requesting you all to please visit at least once whenever you are visiting Kolkata, the beautiful city, the city of joy, and with the Sandesh and Mishti Doi, you can have a taste of our sister Nivedita University. Okay. So the Sister Nivedita University is a very, a new university, although, but still, we all faculty members and staff members and high officials, we all are trying, and with my lovely students, we all are trying hard to put this university forward and to go ahead. And I request you all that please do visit once at our university, at our department. We five faculties are there with me, Rajat Pal. There are four different other faculties. Professor Thakur Chaudhuri, who is going to speak today. Professor Calcutta, who has spoken in the first day. Professor Atre Ghosh, she will be speaking on the next day, tomorrow. And Professor Milan Boni, he will be speaking on the last day of this webinar. So we five are the faculty members of my department, and we are blessed to have Professor Dhrubajyoti Chattopadhyay as our Vice Chancellor. He is also from the same background, from microbiology and biotechnology background. So under his guidance, we are trying to run our department very smoothly. In our department, we generally offer both postgraduate and undergraduate courses on microbiology and biotechnology, as well as we have started our research for, offering the PhD degrees also. So this is nutshell of my department. Without wasting any more time, I will, invite Dr. Mohit Mojumdar. Dr. Mojumdar, are you there?
Hi Rajat, can you hear me?
Yes, yes, Dr. Mojumdar, welcome, welcome. So, to my all the audience, I would like to introduce Dr. Mojumdar. Dr. Mohit Mojumdar is the Director of Business Development, Fine Biotech. He is actually associated, very closely associated with our department from a long back, and Pine Biotech is also a very close, in a association with us. Pine Biotech offers several courses in bioinformatics. Our students usually avail those courses. And Mohit Mojumdar is a very close friend to mine also, and he is a very good speaker. I have listened a few of his lectures, so I hope that you all people will enjoy his lecture also as well. Professor Thakur Chaudhary's lecture you enjoyed. So I'm pretty much sure that Mohit Mojumdar will give you a very good knowledge about the bioinformatics, what the bioinformatics is. As you all know that in today's world, without computer, we cannot do anything. From the morning to the night, whenever we are waking up from the bed, and still whenever we are going to the bed, everything is done by the computer. Somehow we are related to computer. No matter how much educated I am, no matter how rich or how poor I am, whatever it is, what the earning process I am doing, everything is somehow related to the computer. So definitely biology will not also be apart from that effect. So biology will also be somehow related to the computer science. And when these two subjects, two sciences, biological science and computer science merge together, that forms actually the bioinformatics. I think Dr. Mojumdar will speak more about that and he will explain more nicely. So without losing any time or without wasting any time, I am just requesting Dr. Mojumdar to start his lecture. Dr. Mojumdar, please.
Thank you, Roger. Thank you so much for this kind introduction. And thank you, Dr. Thakur, for this excellent talk. And I think it actually makes sense in the context that what I will be talking is very much related to his talk. So, yeah, as Roger said, that let's not waste more time and start my presentation. So let me just share my screen and please let me know if you can see my screen or not.
Yes, yes, Dr. Mojumdar, it's perfectly is visible.
Perfect. So hello everyone. Welcome to today's session on Introduction to Bioinformatics. So before going into the session, I just wanted to give you an introduction about biology, as already Rajat said about the aspect of computational studies, aspect of data generation, how it has changed biology. So biology as a data science. So biology is rapidly acquiring the character of data science. Billions of data points on genes, proteins, and other molecules are compiled in large files and systematically studied. So, analyzing this data will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crop, livestock, like it affects almost everything. So, my name is Mohit Mazumdar. I'm one of the global representatives of Pine Biotech. Pine Biotech is a bioinformatics company that is working towards providing cutting edge research and education for students and researchers in India, US, Africa, and around the globe. So, our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform. We work with students and researchers, provide research support, big data analytics tools designed for biomedical discovery. So, ultimately, we are working for a future where we can contribute to enhance human health and well being by enabling biological research and discovery with data solution and support. So I'm really honored to have been working with people that have a diverse experience in this area, including our CEO and co-founder Ilya Brodsky and other co-founders of the company, Dr. Alfred Trauber, who is a philanthropist who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. So he is the core of, he started the Trauber Bioinformatics Research Center. It is one of his projects. So at another co-founder of us is Dr. Leonard Brodsky. He's the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics and over the course of his career has developed multiple novel algorithmic approaches for the analysis of biomedical data analysis. So these methods have been widely used. On the right, you could see Julia Panoff, Avi, me, Joshua, and Deepsha. We've been working to develop this platform into a educational resources, provide educational resources for students, collaborating with scientists around the globe and helping them with their research. As I was telling you, we are a US based company working closely with several universities across the US, collaborating with Tauber Bioinformatics Research Center and developing these tools and programs and courses like Roger said, we have already have a collaboration with SNU, and we did this data science for biologists course last year, which was very successful, and we hope to develop more collaboration and learn more about the students and the needs of the faculty. Coming back to today's topic, today we will try to understand the application of data-driven biology and application of bioinformatics in various area of research. So I saw that you guys have just finished this lecture, which is like the core of. I mean, it's there is no running away from learning the basics, but there is a vast importance of what is happening today in terms of translational sciences. You already have learned that how the new science policy or the new educational policy has changed, and we are now more focused on developing solutions using science. It's just not doing the theoretical science, but also applying this science into discoveries, applying this science into products, applying this science into pharma. So there are several opportunities that I today will be talking about. So to start with, I just wanted to say that bioinformatics skills are required for anyone seeking to participate in advanced life science research projects nowadays. And if you see like how it goes, like you start from like when you start learning about science, it's a long journey. That's for sure. A research is something that you develop over the years. But I mean, what is the right time? Shouldn't it be good if you start like really soon, so that you reach that level where we require you to do translational stuff, where we want you to work on vaccines, where we want you to work on and like contribute to the society by working on like different problems, for example this pandemic itself. So a big challenge for those interested to get into bioinformatics or data sciences or the data driven biology is to get started. That is a big challenge because it's like it's not standardized here. So that is what we are trying to do here. So once you get started and getting involved and feel comfortable using basic tools, you can easily continue your journey navigating through complex and dynamic world of bioinformatics. So before talking about bioinformatics or just generally about sciences, how this has evolved into the field that we are seeing right now, because of what has already been happened. So if we recall, it has been 70 years since the structure of DNA was discovered. 50 years since have been since the last time sequencing we did the sequencing. So it has been like a long period of time where we have been developing and learning about all this technology and all this information that we talked about. Dr. Thakur talked about amino acids and nucleotides. So how we learned about those, so it comes through a long journey, but today we have a lot of information. So in 2015, 1000 genome project brought a significant number of whole genome sequences into the research community. And what is this whole genome? What is uh, how this technology works, I will be talking about in today's class. So as you see, like in 1950s, we have had the DNA structure, and then after 50 years, we had the Human Genome Project. This was a very, I mean, it's a landmark project. We spent almost 3 billion US dollars. If I say we, I mean humanity. So we were hoping that after the Human Genome Project, we will be able to address most of the problems associated with diseases, why this happening, why this viral outbreak, how to solve the issue with vaccine development, and also drug designing and development in general. But what we realized is that the science is way more complex than what we think it is, because we thought that knowing the sequences will do. But now we understand that the sequences have a really complex role when you put together in terms of how they operate within the organism or within a host or within a specific cell. So how these technologies are helping us understand this complex nature of cell and the complex nature of dynamics of the cell. So there are multiple ways we are generating this data. Okay, so omics data can be generated using NGS, and this technology explores the roles, relationships, and the actions of various types of molecules that makes up the cell of an organism. So NGS, or what is also known as high throughput sequencing, to start learning about this type of big data, the opportunity and the challenges they come with, we will be showing you the resources that are associated with them. But to understand the terminologies in itself, phenomics, genomics, epigenomics, transcriptomics. So what are these technologies? Phenomics is something that we observe. For example, if I give you a really clinical example, when you go to a doctor, I mean, when you visit a doctor, the doctor asks you about your age, right? And about maybe checks your weight. So those physiological factors affects the drug you're taking, right? That's the reason he's asking. He's seeing that whether you're a male or a female, and then also that classification is also something that helps in the diagnosis. So how this technology has evolved into something more deeper, and it has to be deeper because these diseases are way more complex than we thought. So we don't have a vaccine for HIV yet. The virus replication rate is really high. We don't have solutions for all the cancers yet. So what is the reason we have not been able to do it? The reason is that because we don't have the information, or the all the information that is required to know how things work inside. We are gathering information from years now, decades, but the information that is required to understand the whole picture is something that we have started to do now. So the data, as I was talking to you about phenomics, phenomics is a study of those phenotypic characters or phenotypes, as the word that you might have heard a lot. Genomics is about looking into the genes, the sequences, and how it affects. Epigenetics is a study where we are looking at DNA modifications. Transcriptomics is something when we are looking at gene expression. So, there is a state where a person is having, there is a normal person and there is a person who is having cancer, right? So what we do is that we collect a sample from a normal person as a control, and the cancer patient, from the cancer patient, we took some samples, and we see how the proteins are expressed at that point of time, like a person having cancer, a person not having cancer. So when I'm saying proteins or genes, so 20,000 genes in one sample, one person, and like another 20,000 genes in another person. So what we are trying to do there is we are trying to compare and see like which genes are expressed, like highly expressed, which genes are significantly less expressive. So by that we will be able to understand that the targets, or maybe the genes that have been the proteins that have been changed their expression, and why. Those are the questions then we ask, and then we deep dive into the problem and identify that okay, this is the reason. So that is the kind of research that is happening right now in around the globe, and that is helping us transforming science in and developing that understanding. So before, there's a hands-on aspect to every webinar that we do. So the hands-on aspect would be to, go to the site. This is an educational platform that we have created that I was talking to you about, that to help people understand, learn about these technologies, which are not so easy to learn because of a couple of challenges that I told you. One of the challenges that, you think bioinformatics to be something to do with coding. Many people, the courses starts from understanding of coding, so we eliminate that problem because we feel that the biologist who is working on the experiment should be able to understand his own data, should be able to understand. So you are the person who understands, central dogma. You know that you are doing an experiment, what kind of data is, coming out of that experiment. So you should be the one who is analyzing that data as well. So to help achieve that, we design courses. So I just want you to go to this website now and register and take one free course, Introduction to Bioinformatics, which I'm asking you because it is just a very basic course for everyone. It does not need to you does not need to have any knowledge to do this course. So maybe I can also paste a link here in the chat. Just 1 second. So that So if you can see my screen, you just have to create a profile in this site, and then you have to, go and take this course. So this course talks about, introduction to bioinformatics. So I'm just gonna tell you more about it, but just as for a hands-on exercise. Uh, okay, there you go. So please visit this link and take this course. It might just take you 2 hours to finish this, so you can do it afterwards. So as I was telling you that the components of this course, so this is an introductory course that you can take. It's free, and it usually tells you about 2 to 3 things. The course includes introduction to big data bioinformatics, bioinformatics and healthcare, and translational bioinformatics. So you will be easily able to see the application of bioinformatics. Maybe also in agriculture, you will see the role of bioinformatics in defense. You will see the role how this bioinformatics is being applied in translational medicine. That is a very how pharma is using it, how what are the job opportunities that we have right now with these sort of skill sets. So it's not so distinct past where data generation was the bottleneck. So in the past, we were trying to understand by doing single experiments. So we were trying to understand a single enzyme. We were trying to understand about a single protein. We are trying to understand about the function of a single gene. So the structure to function or sequence to structure research is something that is what we are reading in our books right now. All the books that we have been reading about biology, about sciences, has been evolved through that kind of research and have come into the textbooks because of that. So in the current world, uh, things have changed. But in the distinct past, what I was saying is data generation was a bottleneck, and now the bottleneck has changed. The bottleneck is now data mining or extracting useful biological insights from large, complicated data sets. In the past decade, technological advancements in data generation has changed this entire thing. In particularly NGS technologies have allowed researchers to screen changes at varying biological scales. As I was telling you, like how a person is different from another person. So first of earlier we thought that that's like just 1% of the genome, the coding region is what makes all the changes or all the differences, and that's the main thing. Exons, we would just want to focus on exons. Now we understand that that 99% plays a huge role. That no non-coding RNAs and all that research is now, bringing the importance of the other half on the more major part of the genome. So we used to call it junk DNA, but why we used to call it junk DNA, because and still call it junk DNA, because I think we don't understand the way it all works. We don't understand many things, or we are not able to correlate also many of our capabilities to the genome or to the molecular level. But how we are reaching there? We are reaching there by now collecting data. And how we are collecting data, as I was telling you about, time point. So if you get sick in 3 months, you collect your sample and you collect your data and you see yourself, and then you get well, and then there's a series of events that's happening with one individual. So if we collect data on all those events, we will be much more aware of what is actually happening, maybe, because we will be able to compare stuff. We will be able to see that this state one, this is state two, so things like that. But I mean, easier said than done. So because it's a big data, so we are talking about 3 billion base pairs. So genome-wide genetic variation. We are talking about those point mutations that are changing everything. Those point mutations are causing diseases. Those point mutations are, changing the characteristic, one particular trait. Gene expression, I already talked about gene expression. And then small RNA, I talked about small RNA, epigenetic modification, protein binding motif. So all those things, like the chromosomal conformation in high throughput, is something is worth looking at, is worth understanding, because it will definitely gonna solve a lot of challenges and a lot of things in the future, and it's coming up really well. So if you go into this course, you will see that there are videos that explains some of the things that I've talked about today, and there is a quiz. So once you're done with it, you have to finish the quiz. Introduction to bioinformatics can be completed like in very small time. Quizzes help reinforce the learning material, summarizes the topics that we covered. So you can also use like glossary of terminologies, like you can click here, then it will show you what it is. So it's pretty easy, and something to check out. Now, I was talking to you about data-driven discovery. So data-driven discovery involves several steps. So in terms of, thinking, like just thinking about research. So when a scientist does research, when your professor does research, when, all around the globe it's happening in the hospitals, where do we start from? Have you ever thought, like, where do we start from? Does that happen? So it happens through knowledge, because first we have to learn about one thing, that what is out there, before trying to do something new. So if you are trying to do something new, you have to know what has already have happened, because or else you end up doing the same thing that has already have happened. So literature review is very important in that aspect, and knowing what is done and what can be improved is something that you can start thinking about. That's how the research aspect of things work. Like, we know that, okay, this is a problem. Now how do we find solution to that problem? So identifying the question is very important. Review finding, design model, gather data. So what we do is that we look into a problem, and we see that for example, COVID-19 has generated in 1 year so much data that no disease has ever. Like, it's everywhere. Every country, there are bigger labs working on it. They are generating data, and now we have so much data on COVID-19. Now we think that we still need a lot of data, because coronavirus was not well studied before. So we started doing it now. but yet, I mean, we don't have a solution yet. Why you think? I mean, with all this people working and all this information, why you think that it's has haven't been happened yet, and why we are taking this time? So this has been the challenge in biomedical industry. Challenge of doing drug design. Usually, it takes 15 years, 20 years to design a drug, and that was because we were using a lot of high throughput, not sequencing, but screening. So we were trying hit and trial kind of a thing, that whether it works or it doesn't work. And then there are side effects. So when you're talking about a targeted study like this, then it makes more sense, because individually we are all different. So we should have a personalized, personalized way of handling and dealing things. So as I was saying, that every one of us are different in the sense that we perceive things differently. Our body at a molecular level is different, and also the environment that we go through, like the microenvironment, the metabolome, will does also play a very significant role. So how do you understand all these things and think in the context of one person is a thing to be seen in the future, and it's coming up. So really exciting times in that context. So from data to discovery, researchers, are generating large volumes of biological data, which has changed the way the biomedical research is done. So the volume and heterogeneity of the data are limitations right now, that that is challenging our. That is the challenge, and machine learning is playing a role in here because of this. So why machine learning suddenly becomes so much such a big thing? Why data science? Why people are talking about data science? Because you see, like everything is online, and we are generating like megabytes of data maybe daily. Like if you are scrolling your phone, if you're scrolling your website, you're posting your pictures, you're buying on Amazon, everything is getting recorded, and every bit of that is data, and it's getting bigger. So challenges for the company is to analyze that data, and they're doing well. They are using that data, using machine learning techniques to understand their users. And similarly, in biology, what we are trying to do is that we are generating data, at least in the developing countries or developed countries, they are doing it quite efficiently. So there are different steps of designing an experiment, but that you can learn more in this course. So I will not waste more time into this, but to see that this is like like a big cycle. So it's just not like just generating data and relying on it. So there are several elements, just not that. Biological interpretation. So once you understand that, okay, the importance of learning what you're doing right now, as in microbiology and biotechnology, is going to be helpful for your entire course of your career. So whatever you're learning right now is very important. Those books that you're going through is going to help you develop that analytical understanding of how things work. So once you are focused on a single problem, then you of course can just focus on that one problem and do more and more about that one problem. Then you're very you're working on a niche problem, then you can do it because you're not reading about everything around it. So that's the way to go about research, specifically to target something very niche, and then work on it, because there's so much out there. It's really gets overwhelming at the point of time, and we lose interest. So the application of this kind of sciences. So as I was telling you that how this is taken out like from samples, but there are databases that are publicly available, freely available for you to go and look into all of that, like the TCGA portal. The TCGA portal has like a lot of information on all kind of cancers. There, genomic data, you will see genomic data, you will see transcriptomic data. You will see that you can compare a normal person or samples of breast cancer, samples of liver cancer, ovarian cancer there, and you can start working on it right away. It's like that. So that has changed. That has changed, which was not happening before. What was not happening before? There was only limited number of people who had this kind of an access and the tools who can do the research. Now, with the repositories being publicly available, and companies like us who are helping people to understand and learn from this kind of research, it becomes really easier for even students to do research and publish. So the translational aspect of this is to be able to find a personalized treatment, as I was telling you, or what is a personalized treatment? So here in this example, you can see what is happening here is that a sample is taken from a patient. So this is a cancer patient. So this tissue is implanted in a mice. So anthemic. So there are different kind of mice models that have no immune response. They're designed for the scientific experiments. So I think it will take me like maybe another lecture just to talk about the mouse models. But let's not do that today. But just tell you that to to understand the effect of drugs, and we experiment on like mouse models, and that is the tissue is being grafted and in this mice, and then we are testing on different cell lines, that how it is on different cell lines when we put this cancer cell and seeing the effect of a particular drug. So we're trying to do is that based on this information, we are doing molecular profiling. So what is molecular profiling? We are separating people based on their molecular profile. So we actually already know, for example, in breast cancer, there is a panel called PAM50. So in that, what is happening is that we know in breast cancer that these 50 genes, or there are some genes in that 50 panel of genes that are overexpressed. So we know their expression from that these are the genes that will be overexpressed in breast cancer, in a specific type of breast cancers. So those genes or proteins are called biomarkers because we already know them. They are the markers, right? So that is some of the application of this kind of uh, mm, uh, technology. So talking about the platform, once you learn all this, you can get a certificate by finishing, and you will have an academic profile on the platform, which means that we have almost 5,000 people around the globe, many from top universities in the US and top universities in India and many places. You'll find people in there. You can talk to them, collaborate if you find them interesting, and you will have a profile of your own when you finish this course and enroll for this course. And you can put your pictures and stuff, and your social media links for communication. So this is the certificate that I was talking to you about. Okay, so if anyone have any questions for on whatever we covered, please feel free to ask. And now I will just build on that what we have discussed and show you the rest of the presentation. So no questions. Very good. Okay. So
Dr. Mojumdar, maybe the questions will come later on in the chat box or by email. So we will definitely
sure, sure,
get
sure,
to you.
sure,
Yes.
sure. Not a
Mhm.
problem. No, I just wanted if there is any question now. So that's okay. I'll be happy to answer those questions. So many of us here, as I was telling you, many of us are here are biologists that were trained in basic biological processes and then specialized in a certain area where we gained both knowledge and experience from experimental data. Many of the examples we learned from information we accumulated came from carefully designing experiments where the certain assumptions were tested. We were testing an hypothesis. The generated data from those experiments confirmed some of the assumptions, and those were recorded as proven hypotheses. For example, from a biologist perspective, we understand cancer as a set of changes on a cellular and a subcellular level, causing a specific clinical outcome. So once we design an experiment to check for an expected change, we can start associating the molecular change with the phenotype, a clinical manifestation. Ideally, we hope to map out those relationships that we found and make sense of them. So this is a typical way of data connection between the data, and when you trace it back to the pathways, molecular pathways, and you understand identify the key players, identify the pattern, and learn how to manipulate them to find a cure or cure for a condition. That is the challenge, and that is what we are doing in this. So in reality, however, many biological phenomena do not play out according to this formed rules and formulas, because that's what I was telling you, that it's way more complex because of the involvement of multiple factors. But now, after the Human Genome Project, before that we were doing the single experiments. Now, after the Human Genome Project, at least now we know better. So the big data. So when I was just talking about the big data, and it's really difficult to visually or to understand how big it is, because when you see a really tall building, it's big because you can see it for sure. But when we are talking about data stored in drives, it we don't even realize like what it is. We just maybe underestimate it. But it has a few qualities, five qualities that define the big data: volume, velocity, veracity, variety, value. So which is ever growing. So a biologist is largely, driven by a hypothesis, which is often a result of past observations. These observations give rise to a question that can be identified. So it's just like a summarization. I'm summarizing the aspects that we discussed today. So the observation give rise to a question that can be definitively answered using data generation generated in a experiment. This hypothesis can be confirmed or refuted after you analyze the data, and you have come to a conclusion whether you want to support the hypothesis or refute the hypothesis. So the collection of results are knowledge. But big data is changing this way we view results, as I was telling you, that because what is happening here is that for example, this part is already done. For example, like people have generated data, they have deposited it in NCBI or in TCGA, all those repositories. Like NCBI has humongous amount of data, like insane amount of data, but not that all the data has been analyzed, because some people have generated data for a different aspect, maybe for a different biological question to answer, but that data can be used for answering another question as well. So NCBI, as I was telling you, has grown exponentially in the past few years. Just to tell, the recent growth in high throughput sequencing of cancer genomics and transcriptomics has, created a lot of big data problems. So some of the resources and repositories are like this: Array Express, TCGA, as I was talking to you about. So where you can get sequencing like RNA-seq data. So big data and precision in oncology. And I was telling you that identifying a patient and patient stratification is something that we discussed. I know these are like big words, but these are simple to understand. If you go through maybe a learning experience, take the introduction to bioinformatics, you'll understand that this is something that you may be able to comprehend. So the big data problem preludes many cancer biologists and oncologists from gleaning knowledge from data generated regarding the nature of this malignant processes, as well as the relationship between tumor genomic profiles and treatment responses. So how a person is responding to a treatment, I mean, if a drug is given, then if we are looking at a genomic level, that how it is, helping in targeted cancer therapy. So, talking about this therapies and how this entire paradigm has changed, how the pharma is using this, how this big pharmaceutical industry, the small biotech companies are working. So, genomics is now considered as a legitimate big data field. Just one whole genome, human genome, produces approximately 200 GBs of raw data. So one genome is approximately 200 gigabytes of raw data. So if we manage to sequence maybe 100 million people, which is happening in many countries, so 100 million people by 2025, we will have what? We will have 20 billion gigabytes of raw data. So that's massive, right? So when you have this sort of data sets and databases, you need maybe a computational infrastructure to be able to, or be, even to understand how they are connected. So, just to say, like acquiring bioinformatics skills is in today's world is of tremendous value, because if you see the postdoc positions that are being currently offered, or how industry is being evolved, I think a lot of job opportunities are there. But you probably know that the online courses certificates are not yet valued that much. I mean, one of the reasons for this is due to the lack of maybe community oversight and mentorship. How many times? I mean, I have I have this experience. So how many times? I mean, I have gone through Coursera edX and felt sleeping watching a video. So I think one of the things that is with this online way of learning is to be able to interact like the way we are doing it right now. So once we are doing it through like fairly regularly, I'm very sure that you'll ask more relevant questions and understand this into more depth. So I think this hybrid way of learning is really nice, and that's why we have so as I was saying before, that these courses are free. So Introduction to Genomics is a free course, and as you have learned in the previous class, every aspect of central dogma, and then what is DNA, what is ATGC, and how these mutations are causing are happening, and the variation, like why we are different. I mean, why how a mutation plays a role in that. So, Introduction to Genomics is a course which will help you visualize genome chromosomes on NCBI browser. So it will be, you will be asked to open another browser, and there will be a file which you will click, and it will show in the browser that how you can see this entire genome, the chromosomes, and the segments of genes in that. So please, it's a free course. I'm recommending you to take it. Introduction to Metagenomics again is another free course, which covers metagenomics. So microbiome and metagenomics, how they are playing a role. So I talked about environment, how environment plays a significant role in how we are reacting to different this maybe in terms of medicine, how drugs are affecting us. So to understand that, I mean, people are studying the role of gut microbiota. So how the samples taken from our gut and the mixture of bacteria and all the different microorganisms are there can help understand more about our body and how it influences our body. So human gut American gut project is a project that happened where they've taken samples from a large community of people and did that analysis. So it gives you an overview. So this is again a free course. Introduction to Epigenetics, to learn about epigenetic regulations and changes and DNA methylation. So different type of all these changes that are happening within the cell, within the DNA, which is not a permanent change. So to learn about epigenetics, transcriptomics one is not a free course; it's a hands-on course. So when you do transcriptomics, I mean, after you are like well versed, and if you find this thing interesting, so in transcriptomics one, what you will learn is that the gene expression. We're talking about cancer patients. We talked about cancer and normal cell. We talked about the PDX, the mouse project. So in transcriptomics one, what we do is that we learn about what is transcription translation, why we learn RNA, why we want to learn about RNA, and how we can quantify RNA. Like from before, we used to use northern blotting and microarrays to understand and quantify RNA. Now we are using NGS technology and getting the gene expression. So I gave you an example. So that example is being covered in this, in the context that it is application of what you have learned, plus you have assignments, you have hands-on tutorials to work on the T-Bioinfo server. So the T-Bioinfo server is a cutting-edge platform designed and developed for students and biologists and clinicians. And why I'm saying this is that because maybe I can give you a demonstration. So is it visible, Rajat? Is this page visible?
Yes, yes, Dr. Mojumdar, it's visible.
Thank you. So this is the T-Bioinfo platform, and this is an intuitive platform, which means like when if you want to learn that how things, it looks really complex, and it is complex, but we want to help people understand this complexity easily. So to do that, I mean, for example, I will give you a small example today about. Maybe I have to log in first. So this login is not for everyone, but for people who are a participant, or like if you are working with us, something like that, on a program or something. So however, this platform is free to use, like the introduction to bioinformatics. So this is, for example, this, you can say that this is our books. So this is my book, and this is the workbench. So this is where I do the experiment. So to learn about these experiments, I'll just show you a demonstration pipeline. So this is the cell line project, and you can see this is a complex bioinformatics, workflow. It has a lot of algorithms that do different things, and they are placed in a logical manner, so that when you input the data, it goes through this process. So it is first pre-processing, and then it is cleaning. The cleaning stuff is going on. Then we are aligning it to genome or transcriptome, and we are looking at the isoforms. So I did not discuss about the isoform problem today, but that would be another session. So what we are doing is, here we are taking samples from a cancer patient and normal patient, and we are putting it inside this inside this workflow, and see how we can get the table of expression. So what is happening, what happens in NGS, when you do this experiment, NGS experiment, you get a raw reads. You don't get like table of expression. So from raw reads to table of expression is a complex experiment. So we can do it easily here. And why I'm saying easily, and it's meant for everyone, because when you click start, yeah, I mean, it shows like what is there, and it's a point and click system, so you don't have to code and do all those things to get this kind of experiment running. So on also, this is an AI guided platform. So what is happening here is that when I click start, it has highlighted me a path that based on this data I should use Trimomatic. So Trimomatic is something, is an algorithm that helps us to remove the technical sequences. So what happens is that in sequencing, in NGS sequencing, when we are putting that sequences into the sequencer, we are we are uh, joining adapters to the sequences. So those adapters are like sequences that comes and goes and ligate at that like 5' end, and those are the technical sequences. Those are not our sequences that is coming from the samples, but those are for the experiment to happen successfully. So to do that afterwards, like after when you get the raw read, it will come up with all that noise that have been that is the limitation of the experiment. So we are not getting a pure result of sequence. So we use a lot of statistics and mathematics and algorithms from that state of raw data to a biologically meaningful data. So that is the whole process that you will learn in transcriptomics 1, that how we will be able to solve and understand transcriptomics data analysis. So that is one of the aspects of transcriptomics data analysis. And this is the entire process. So once you are generating a sample from a sequence or COVID patient in Kolkata, for example, you have the genome. Now what to do with that genome? I mean, you have to clean it up. I mean, it's coming from the machine, it's a raw, and then you use platforms like this, or you develop a platform where you can do this sort of analysis. And then once you have the entire genome sequence as a FASTA, then you can do further analysis. So in that context, this is a point and click system. As I was telling you, that I will click on Trimomatic, it will tell me next I have to click here, and I will be able to know everything about this algorithm. It's described here, and there is also publications that are also associated, that are also put in here. Just so I will do. So if you can see, there are publications. If the algorithm is published, you can find the publication and learn about the algorithm. Input output files is a demo pipeline. So this is point and click, means that you have to just click and you can go back and change that path and stuff. So once you do that, you have the file as an output. So this is for teaching. We have built this platform and this demo pipeline for faculties to teach, and also for us to teach. So it's what we did in here is a big experiment, but we will be able to understand through like different sessions and understand that what is this isoform expression, what is this gene expression, and how we analyze this and understand whether they will be biomarkers for cancer, whether they will be a drug target for cancer. So these are important, very important questions in terms of biological research, or individual research, whether it is in agriculture, whether it is in defense, or whether it is in different aspects. In understanding the mechanism of an enzyme, also, for example, not an enzyme, but mechanism of a disease, mechanism of a behavior. So there are multiple factors involved. So we use machine learning for that. So that I think we will not be able to cover in this one, but you can obviously learn all that using the platform. So as I was telling you, that this platform is developed by Tauber Bioinformatics Research Center, Haifa, Israel, and we have several collaborators in UCSF, Cambridge, that helped us, with several of these pipelines to be designed and being able to be used by the scientific community and students. So the state of art design helps us to go through this and do a high quality research in a smaller amount of time. So that's one of the benefits. And machine learning is something that we use for the big data, because it's very difficult to understand the pattern. If you, for example, you can plot two genes. I mean, you can plot, like, for example, mm, uh, age. It would be better to give you a visual, so maybe I will skip that. So just to give you an example, like it's easier to plot data into XY and XYZ. So two-dimensional, you can plot data and understand analysis. You can plot three-dimensional graphs and understand the trend. But when you have thousands, when I'm talking about thousands of genes, then it becomes a problem of dimensionality. So a problem of dimensionality is that it's not human readable. You cannot plot 4D graph or 5D, 6D, 7D graph, but machine learning can. So why machine learning is helpful is because this huge data, from this huge data, machine learning is able to give us the important data. So to the understand the variation. When we are comparing, for example, a normal person with a cancer patient, we are comparing. Always remember that we are comparing, and when we are comparing, we have to compare the entire both of them together, and that's a computationally a challenging thing to do when you have not hundreds, not but thousands, and maybe more than that of number of samples. 100,000 genes you have to look, and you have to find maybe 20 genes out of it. How would you do that? You would take help from machine learning. So, as I was telling you, we just did the course previous year, and we are doing different online programs to help students and our universities, collaborative universities and research centers. So this online programs, the Omics Logic programs, have three components. We do Zoom meetings and this asynchronous courses that I showed you. So when you take these courses and you get also points for doing that. So you will see that how much you have progressed and how much you have learned, and also you get certificates that gets stored in the profile, and that could be shared, and it's always there, so you can put it in your LinkedIn, or you can show it to your recruiter or whatever. So another aspect is the practical assignment. So we have come up with a Python and R tutorials and courses, and it's in a similar way we have designed a platform so that people can easily learn Python and R and implement those kind of skill sets as well. So I would talk about a couple of programs that we do. Omics Logic Transcriptomics is a 1-month program, which we are going to start on 16th September, where we will talk about entire transcriptomics, data analysis. So transcriptomics 1, transcriptomics 2, 3, 4. We have four courses. One talks about the entire, raw data to gene to the table of expression. And when I say table of expression, it means the genes, the list of genes that have the expression levels in numbers. So you can just read in numbers, see in numbers that how a gene is more expressed and how a gene is less expressed. So in transcriptomics 2, what we do is statistical analysis. Transcriptomics 3 is machine learning. Transcriptomics 4 is single cell transcriptomics. So that is again another advanced, aspect of learning how to interpret single cell data. Metagenomics, again, about the microbiome and about understanding how these viruses, how these small organisms are affecting the microbiome and affecting different cellular processes. Genomics is to understand and learn about genomic data analysis. Epigenetic, I have already told you about. So, other than that, like the Omics Logic programs, we have like 4 Omics Logic programs you saw on data types. So those are data types. Now these are research programs, which means that we are addressing a specific research problems. Like for example, infectious diseases. We did a program where we learned about when we did a coronavirus, we took all the sequences of coronavirus sequence, and we tried to address many biological questions. And this was a really successful program that we are doing with students from all around the globe. People came up with bioinformatics projects from coronaviruses, different type of SARS-CoV-2 strains. People from Brazil, they came up with their own. Sri Lanka. So like it has been an amazing experience. People working from different outbreaks. So we are here analyzing data from different outbreaks and understanding the biological significance and what can be done. Precision oncology is a specialization track for precision oncology. Precision, which means that cancer. We're talking about different cancer. If you're interested in that, you can check this out. And bioinformatics for precision medicine is about precision medicine, which means that addressing the challenges and future of medicine, how this knowledge can be transformed into drug designing and understanding how we can have personalized, techniques to help individuals. We did a course on SARS-CoV-2 specifically on just for SARS-CoV-2. So anyone who's just interested in learning all the aspects and understanding how the spike proteins and how the sequence from the sequence to structure to function. And this is a course again for data science. This is for statistics, Python, R. So these are specialization course, or you can say advanced courses that helps you develop skill sets that are matching to the industry right now. So the CodePlay, this third platform that we are launching this month, is for Python and R. So you can just put the code. Every help is there. You can run it from within that and generate visualization stuff. So when we have like these projects, when we have the students finishing these projects with the university, for example, the students that finished the courses in SNU, we offered them fellowship or research internship where they learn about or where they progress on a specific problem. So you learned about the data types, you learned about the examples where it has been implemented, you learn about the research, you learn about how it can help in translational kind of science.
So as I was telling you that after we finish the after one finish the program, like faculties or students, and they are whether they are interested in research, they are interested in developing curriculum, to students interested in developing research projects. So once you have learned about the data types, once you have learned about the examples, once you have gained enough points on the server, then we invite people to work on research projects, and that is a bioinformatics fellowship program that we do. So other than that, that is another program, that is a fellowship program, that is for people who are like maybe in an advanced stage, like in third, fourth year, or maybe like doing a research project, or after learning everything, they want to work on their independent research project or have their own idea. In that sense, that research fellowship is helpful. So these are the some of the areas that we are working on: infectious diseases, data science, precision medicine. So this is programming and coding, precision oncology, so cancer, neuroscience, and metagenomics. So these are the research areas we have developed specialization tracks for students to learn. So research internship helps you develop, understand some of the concepts. For example, going through the omics data, learning about the omics data, mastering the T-Bioinfo platform, which I showed you, mutation variants, gene expression, metagenomics, biostats, machine learning. So this is all in, in a sequence, because first you want to learn about the basics that you're learning in your college by your beloved faculties and very talented faculties. So once you learn from that, I mean, it would be nice if you can, understand some of the modern biology and the applicational biology and translational aspects. So these are some of those things that will help you understand. So as I was telling you, the people who finish, they do go on and publish, present their posters in conferences, and talking about what they have learned and how they have used this for their own research. And another aspect of it is to publish. So poster publications we have had from our so this is why I'm showing you this, is because this is an outcome that is coming in maybe 3 to 6 months. That is literally very difficult to achieve for any institution. But with the help of these platforms and this very niche kind of an understanding, we were able to generate and people were able to finish bioinformatics projects and publish posters also in like F1000 research, research gate. So these are some of the benefits. And once you have well versed and done, you like these are some of the candidates. Hannah is from high school, so we have same programs from high schools to PhD and to faculty and to researchers. The same platform can be utilized by all of people needing to or have that enthusiasm to learn. So the most important thing is to be able to have that research interest. And being a biologist and microbiologist in SNU, I think it would be a really nice thing for you to start looking up and look up for these keywords, learn from your professors. You can reach out to us, so you have the opportunity that is a rare opportunity for anyone else. So please utilize that. And after you're done with all of it, and if you're confident, we offer jobs. So we have already hired three students. So the jobs are, to develop curriculum or to train in omics logic programs where we take sessions like the omics logic programs I've showed you, and also to help students. Like all these programs have a component of mentorship. So the interns that we have are helping the students of our different programs. And now with the help of this collaboration between mentors, students, faculty, this entire process is very highly effective. So I will just end with this slide, and I'm open to questions. So please go ahead if you have any questions.
Yes, thank you very much, Dr. Mojumdar. Um, do we have any questions? Just let me check if we have any questions here or not. Maybe not in chat box or through email. We can have some questions.
Sure, sure. Please.
Anyways, whenever some question will come, we will communicate to you to address those questions. And you have explained a lot about the programs offered by your company and how the students can take the bioinformatics as their career. So it's a nice talk, Dr. Majumdar. I'm really very glad, and it's a very big opportunity for us to have you, a director of a company. And definitely, my students are now very much. I mean, they are a little bit of worry before that where we can go, where we can have our jobs. So they are now relaxed that we have fine marks for job opportunities, and Dr. Mojumdar is there. Okay.
Thank
So
you, and I hope that this is helpful. And thank you, Rogers, for inviting me, and thank all the dignitaries.
yes, yes. Actually, in online platform, we have to face some problems. Sometimes the net problem is there. Yesterday also in my session, the net got disconnected, and I was talking on my own way. So I beg pardon from all my participants that we are helpless. We cannot do anything. So please, forgive us for these kind of difficulties, technical problems, and be with us for next 2 days again. We have more sessions, good talks we are going to have again. So, is there nothing to more discuss? May I conclude the session with the special thanks to our two very eminent speakers, Dr. Mohit Mojumdar from Bain Biotech and Dr. Shomshubha Thakur Chaudhary from Sister Nivedita University. And I will thank to Fatima Calcutawala, Professor Atrik Ghosh, and Professor Milan Boni for staying with us and helping this event throughout this 5-day program. So, okay, so I will conclude this session for today, and all the participants hope to see you tomorrow again on the same platform at the same time, and the link again will be sent to you by tomorrow. Have a nice day, stay safe, stay healthy, and bye for now. Goodbye, everyone.
Thank you.
Goodbye.
Bye bye.
Goodbye.
Yes.